So chapter 17, special senses. Learning outcomes, you guys can go through it. It's basically what we are going to be doing, learning outcomes. And these are the special sensations. Olfaction is also known as smell. You need to know both. Gustation is taste. Vision, equilibrium, also known as balance, and hearing. Those two usually come together because this is the function of the ear. We do the hearing and equilibrium in the ear. So this is two, and we have another three. That will be the total of five. That's why in the movies they say the sixth sense. Like this is five, there is an extra one predicting what's going to happen in the future. I don't think so, but they say it. So the first one is olfaction. Um, so olfaction is happening in the nasal cavity, basically, and we have olfactory sensory neurons. There are receptors in our nose on the top that are going to detect anything that we are smelling. Okay, how do we detect it? As we go, I'm not going to go exactly word by word, just to let you know. Okay, if there is something that's not very important and very critical, I will go through it fast. When there is something important, you will know. Okay, so something like this. How do we detect the smell? There are chemicals, dissolved chemicals, that are in everything that we are smelling, whatever it is, food, flour, anything that you smell, there are dissolved chemicals. And these chemicals are going to go and interact with our receptors, stimulating them, creating the sensation. That's not only here for the smell or olfaction. Taste is the same thing. Both of them dissolved chemicals. They go and interact with our receptors on the top of the nose, creating an electrical signal that goes to the brain, and the brain realize what it is. Okay. Aside from that, there are other functions of the nose besides smelling, obviously breathing, olfactory glands, these glands that secrete secretions for cleaning and trapping and so on. But the most important thing here is how do we smell? Dissolved chemicals. So look at it. These are the receptors. And like this, how do you smell the flower? Dissolved chemicals goes into the nose until it reach the receptors, olfactory receptors. They interact with the receptors, making an action potential. You guys remember the action potential from 233, which is electrical signal. This electrical signal is going to travel as follow, and this is also important. What is it? Receptors, nerve fibers. The nerve fibers are going to cross and go inside the skull. How do they cross? Cribriform plates. You guys remember the cribriform plates in the skull that have a lot of perforations? The nerves or the fibers travel through those openings until they collect in this part, which is the olfactory bulb. After the olfactory bulb, olfactory tract. Both of them combined, olfactory nerve. So it's two together, and the pathway is important. From there, it will travel all the way until it goes to the olfactory cortex, okay, in the brain. So this is the inside of the nose. You also have these elevations, three conchi or conchi and three metasis. But this part is the most important. Receptors, cribriform plate, fibers go through it, bulb, tract, until it go to the brain. Here it is again. These are the receptors. Dissolved chemicals, again, stimulate the receptors. It goes through the fibers, travel through cribriform plate of ethmoid until they reach the bulb tract and so on. So what happened when the dissolved chemicals What happens when the dissolved chemicals interact with the receptors? Specifically, it binds to a G protein in the receptor, and this will generate action potential, which is depolarization. This depolarization is going to travel from the fibers to the bulb to the tract until it goes to the brain. And we call that the olfactory pathway. It goes, what's the final destination? Olfactory cortex. Is that pathway important? Of course. I wouldn't focus on it if it is not. As it goes, it stops in by the limbic system. And there are some students here that attended my 233. So, but I will make sure to give you guys these mnemonics and stuff that you can use to remember. For the limbic system, I always say that the function of the limbic system is limbic contain M. It should remind you with memory and emotion. Memory and emotion. Okay? So what this has to do with smelling? Don't you feel something when you smell flower versus something bad? Don't you feel? Right? Yes. So this is why it's stopped by the limbic system. So you have this feeling. You feel good, you feel bad, and so on. So this is for olfaction. Gustation will be basically the same thing. Olfactory discrimination, we can distinguish different things depending on the way they interact and the protein that they interact with, which is associated with the receptors. The olfactory receptors do a lot of turnover. We lose it and we remake it. Same thing for the taste. Okay? These receptors, we lose it, we make it. We lose it, we make it all the time, which is called the turnover. We replace it. And then, generally speaking, the numbers decline as we age. So the number that you have is different than mine. Mine is obviously less. When I go older, less, less, just like anything else with age. The next is gustation, and gustation is taste. The taste receptors are located mainly in the top of your tongue, and this is called the dorsum of the tongue. The dorsum is the upper surface. The lower surface is the ventrum. Okay, so like this. This is in the front ventrum, and this is in the back. So the upper surface is the back. Where are these receptors located? In the taste buds. Only in the taste buds? Most of it. Some of it are back in the pharynx. Okay? These buds are located in the lingual papillae. So, receptors in the buds, and the buds are part of the papillae. So, if you look in the mirror, you will see these papillae on the back of your tongue. You can't see more than that. But if you look more like a magnifying lens or something, you will see the buds, and in the buds, there are receptors. Okay? Papillae, buds, receptors. Receptors are located in the buds, which is located in the papillae in the dorsum of the tongue. These papillae can look like any one of those. Filiform, fungiform, velate, foliate. There are 4 different types. It all depends on how does it look like. Okay? The filiform doesn't do anything. It just moves things above the tongue. It doesn't do much. Fungiform, it does have velate and foliate. All of these, they have the taste buds. So the first one, which is the filiform, is the one that does not contain any taste buds. The taste buds that contain the receptors, they also contain stem cells. What are the stem cells? The stem cells are the cells that generate other cells, stem cells. You lose cells, the stem cells will replace it. Where is it? In the taste buds. Okay? And In these buds, in the cells, there are pores. When you eat something, when you drink something, some of these dissolved chemicals go through the pores and reach the receptors and do exactly just like the olfaction. Okay? It interacts with the receptors, generating an action potential. And this action potential is going to travel same way until it reaches the gustatory cortex, which is located in the insula. Do you remember the insula? The insula, frontal lobe, parietal lobe, temporal lobe, uh, occipital lobe, and then the fifth one that's isolated and that's more deeper. This is called the insula. This is where the gustatory cortex is located. So here it is. This is how it looks like. If you look in the mirror again, you will see these because these are the actual papillae. You don't see the inside of it, but you see the papillae like this with different shapes. And there are some in the pharynx as well. What is in the pharynx? Water. Water receptors are in the pharynx, meaning if I put a drop of water in the upper surface of your tongue, you don't taste it. It has to go and goes actually into the pharynx. These are the water receptors located in the pharynx. The pharynx is here, the blue. That's okay. The pharynx is behind. Okay. Um, Otherwise, all of these are different types of receptors. And here, how it looks like, if you are interested to know, vallate or circumvallate, like a circle after circle. This is how it looks like. Foliate looks like a bulge like this. Fungiform kind of looks like a fungus. That's if you are interested to know. But what I actually want you to know, filiform does not contain buds, just move. The other ones contain the buds. Here it is. If you look under the microscope or something, um, these are the pores. The chemicals pass through it until it reach the receptors, creating an action potential. This is important to know. Primary taste sensations. These are the basics. When you eat or drink something, you can say, this tastes like sweet. This is sour. This is salty. This is bitter, right? So these are the primary taste sensations. There are others, but these are the primary that you have to know. What are the additional? Two, umami and water. These are not the primary ones. These are the additional ones. Okay, so we have four primary and two additionals. Umami is that taste uh, by glutamate. It's basically if you're like eating like a broth, chicken broth or meat broth or parmesan, whatever you feel at that point, this is called umami. Okay. Water is an additional one in the pharynx specifically. Obviously, same thing. Just like smelling is different from person to person. Some of us have very strong smelling sensation and others are not. Same thing for the taste. And also, the number of receptors decline. The rule of the thumb is everything decline. That's it. There is nothing that doesn't decline with age. So just take it as a rule. Okay, here is a gustation. And um, we didn't need to go into all the details. This is more of a 233, but just understand that how do you create the electrical signal or the action potential? It is dissolved chemicals interact with the receptors, G protein, and the G protein will do the rest, which is sodium in, potassium out. Do you guys remember this? It's just the action potential, and I don't want to go more into that. Too much details is not what I need. I want you guys to know the core of the chapter. Okay, next is the eye. And in the eye, we have the eye itself and we have accessory structures. These accessory structures are the eyelid, the lacrimal apparatus, and the rest of the epithelium. We will discuss it. The eyelid is called palpebra. You have to remember this. When you see two names, you have to know the two names. Okay? So eyelid is called palpebra. If you don't know palpebra, you will not understand palpebral fissure, palpebral muscle, palpebral. You have to know it. Okay? So palpebra is basically the eyelid, and the palpebral fissure is this gap in between the two eyelids. Upper eyelid, lower eyelid, in between there is a fissure. And there are two angles, medial one and lateral one. Obviously, this is medial and this is lateral, right? Each one of those, if you look in the mirror, in the right angle, in the medial angle and the lateral angle, you will see like a fleshy like bulge in the right and in the left. It's more, I mean, medial and lateral, it's more in the medial. Just look in the mirror, it's very easy to see. And this is called, uh, uh, what is it called? Skipping my name, my memory right now. I will remember it. But also there are openings that are called the canthi, the medial and lateral canthus. I will remember it. Caruncle. It's called caruncle. I'm sorry. It's called the caruncle. And right, and both sides, both angles, you have this called caruncle. Obviously, we have the eyelashes. The tip or the border of each of the eyelids contain glands. It's called the tarsus. So this is the upper eyelid and this is the lower eyelid. This is called the tarsus, this edge. This is the one that contains the hair, okay? The eyelashes. Plus, there are glands that secrete oily secretion to avoid sticking of both eyelids. These are called the tarsal glands. Caruncle is what I just mentioned. Right here, more. This mass. So here it is. This is the medial angle. This is the lateral angle. This is the caruncle right here, the lacrimal caruncle. This is the upper eyelid or upper palpebra, lower palpebra. In between, this is the fissure, which is when you open it, you see your eye. When you close it, you don't see it. So it's in between. Otherwise, we will talk about the rest later. Okay. The next is, we still didn't get into the eye itself. Okay, we're still talking about the accessory structures. So if you open your eyes and look in the mirror, you will see white and a circle of whatever the color of your eye in the middle, right? White, and in the middle of it, this circle, depending on your eye color, it can be brown, black, green, whatever it is, right? The white that you see outside, the white is sclera. We will talk about it. But if I try to touch the white and I'm thinking that I'm touching it, I'm not touching the white. There is a cover outside of it. That's called the conjunctiva. Okay? So what you see is not the superficial that you can touch. You're not actually seeing the white. There is white and there is a transparent sheet outside of it. This is the one that contain the capillaries and the arterioles, okay? So there is a sheet outside of the white, which is called the conjunctiva. This conjunctiva is covering the white, which is the sclera, and then it reflects to underline or to cover the eyelid from the inside, okay? Look at this. This is the eye itself, okay? This is the white. It covers the white, reflect, and then go to line the eyelid. So if I, like some kids do that, right? They flip their eyelids. When you flip your eyelids, what you're looking at is part of the conjunctiva. So there is part of it on the eye, on the white. It reflects and then line the palpebra from the side. Does these have different names? Yes. The one on the eye itself, on the sclera, is called the bulbar conjunctiva. The one that's lining the eyelid from inside is called the palpebral. Why? Because palpebra is eyelid, isn't it? So it in lining the eyelid is called palpebral conjunctiva. Okay, so we have two of those. Conjunctivitis is basically inflammation of the conjunctiva. If you put itis at the end of anything, it means inflammation or infection. Do we know that? It's a suffix, itis. You put it after anything, inflammation or infection. Conjunctivitis, stomatitis, gastritis, anything itis, hepatitis, right? Anything you put itis at the end, inflammation or infection. And I always say that in 233, and I say it again in 234. Medical sciences are all about prefixes and suffixes. Okay, the more you get, it's not English. This have nothing to do with English. English, each word is as it is. This is not the same. This is prefixes and suffixes. You put them together, you get a meaning. So if your first time ever to hear a word, you can guess it. If you understand what's the prefix and the suffix, like conjunctivitis. What's conjunctivitis? I don't know. But conjunctiva is this membrane. But I know that itis is inflammation. So what's conjunctivitis? The first time that I hear about it, inflammation of the conjunctiva. Just because you know conjunctiva and itis. Okay? So I will be talking about different, and I will give you a lot of these prefixes and suffixes. This is going to help you a lot. Lacrimal apparatus is that apparatus that secretes the lacrimal secretion, which is the tear. The main gland itself is right here. Okay? There is a gland that will take it from there and pour it into the eye. The tears have different functions. It's not just cleaning the eye. It cleans the eye, moisturize the eye, and it also contain, can we stop talking? It also contain lysozymes, okay? Antibacterial enzymes, three functions that you need to know of the tears. Remember the two parts of the conjunctiva, pulpar and palpebral, the two parts? This fold in between those two is called fornix. Fornix. This is where the gland ducts open. They open in the fornix. So we have a gland right here, main gland and accessory gland, ducts pouring the tears or the lacrimal secretion into the eye. It cleans the eye, moisturize the eye, kill the pathogens, and then leave. How does it leave? So this is how we secrete it. How does it leave? It leaves through the lacrimal punctum or puncta. This is the plural. Openings, tiny, one above and one in the upper and one in the lower. Then it passed through canaliculi. I will show you the pictures, collecting into the lacrimal sac. The lacrimal sac is located here. Okay, I will show you, and we will see the models as well. So it will make more sense. But after the tears leave the eye, how does it leave? Pores, canaliculi, they collect into the sac. And then there is a duct that take it from lacrimal sac into the nose. The tears go to the nose? Yes, it does go to the nose. We don't feel it. If I ask you, do you know that the tears go to your nose? No, I don't feel it. Yes, because it goes very small amounts. It's like make a tiny film. Of course you don't feel it. But if you cry or if you're making a lot of tears, sometimes you might feel it in your nose. Okay? So it goes actually to the nose through the nasolacrimal duct. Look at this picture right here. This up here, lateral, this is the lacrimal gland. It produces the tears. It go to the ducts. The ducts open in the fornix. It go into the eye, around the eye, cleaning, moisturizing, killing pathogens. And there are two openings right here, very tiny. Exactly, it looks like you get like a tiny pen or something and you make an opening, very tiny. So there's the one here and one there. The superior and inferior puncti. From those, look at those white here, canaliculi, superior and inferior canaliculi, collecting right here, lacrimal sac. From the sac, it goes down into through the nasolacrimal duct, and the name always makes sense. Nasolacrimal duct. The duct between the lacrimal sac and the nose. Nasolacrimal. Okay? What do you mean by canaliculi? It sounds like canal, right? You just add to it eculus. Eculus means small. So canal is something big. If you add eculi or eculus, it will be tiny. Canal, canaliculus, tiny canal. That's if you're interested to know the meanings, which is actually helpful. The three layers of the eyeball, outside, middle, and inside. We have three layers. The outer one is fibrous tissue. The middle one contains vessels, so we call it the vascular, also known as uvea. One more time. When you see more than one meaning, you have to know it. Do I need to know intermediate or uvea? Both of them. Both of them. You don't know which one will show up in your exam and which one we're going to use. If you see something like uveitis, if this is the first time to hear it, uveitis, right? Uvea is the middle layer. Itis, inflammation, right? Uvea. And then the innermost is called retina. The eye is surrounded by fat, just like the heart, just like the kidney. The fat there is not the bad fat. This is good fat. Why? Because it acts as a cushion, like a pillow, surrounding these very sensitive organs. So if you look inside the eye, the eyeball itself, there are three layers. But if you look inside, you will see that space, and this space is splitted into two cavities, anterior cavity and posterior cavity. The anterior one is the small one. The posterior one is the big one. I will show you the pictures. So this is the eyeball. There is a division dividing it, okay, which is the lens. I will show you. The lens is dividing this ball like this. Anterior to the lens, anterior cavity, small. Posterior to the lens, posterior cavity, big. Since these are two cavities, do they contain the same thing or they are empty? It's not empty. If it's empty, it will collapse. The anterior one contain watery secretion that's called aqueous humor. Aqueous meaning water or watery. So the anterior one contain aqueous humor. The posterior one contain vitreous. What's vitreous means? Vitreous means jello-like, something that's thicker. Okay? Look at this. This is the whole eyeball. This is the lens right here. Look at the purple or the violet. This is anterior cavity. And look at this big pink in the back here. This is the posterior cavity. What do we have in the anterior cavity? Aqueous humor, watery. What do we have in the posterior? Vitreous humor, jello-like. Why? To keep the shape of the eye. The eye should be spherical structure. If you don't have it, it will collapse. This is keeping the shape of the eye. For the anterior one, why you don't have vitreous humor, the jello-like, just like the anterior one, the posterior one? Because the aqueous humor in the front, in the anterior, is not only to keep the shape, it's actually to feed the structures in the front. Secretions to feed? Yes. There is no blood vessels here. There are no blood vessels. Nutrients, how you get nutrients? I get nutrients from the blood vessels. No blood vessels. It's avascular. This anterior part that you see in the front, what's A? No. Avascular, no vessels. How can you get the nutrients? Aqueous humor, okay? Look at this violet or purple. It's further subdivided by the iris. What's the iris? The colored part of our eye. You say, my eye is green, black, brown. You're talking about the iris, basically. It subdivides the anterior cavity into two subcavities or chambers. Okay. Uh, are we supposed to finish at 7:10 or 7:15? Does anybody know? 20? Okay, good. Okay, so now we got the cavity, right? Did we understand the cavity? Okay, how about the three layers? Outer fibrous, middle vascular, uvea, inner nervous, which is called retina. Three layers. Outer, it is three parts. The outer is three parts. Sclera, this is the white of the eye. In the front, let's say if you open your eyes like this and you see the white, there is this black in the middle or brown or whatever in the middle. If you touch it, you're still not touching the brown or the green or something. You're touching something that's transparent again. And this is called the cornea. We will see all of that in the lab. So sclera is fibrous tissue. It is white. Cornea is fibrous tissue, but transparent. It has to be transparent. Otherwise, you will not see, right? The eye rays, the light rays enter your eye because this is transparent, which is the cornea. And then there is a junction in between, and here is a picture. Look at this. Look at this. It's blue, but it's supposed to be white. You see this outer one? This is the sclera. And look at this. This is the transparent here, cornea. And this is what you see, iris, the colored part. You see the colored part because outside of it is transparent. Okay, so what's the sclera plus the cornea together? This is called the fibrous layer or the outer layer. The next one is vascular because it contains blood vessels. Okay, what are the different parts and why this vascular layer, why it contains blood vessels? Because it's feeding, it's in the middle, and it contains blood vessels in the middle, so that it feeds the outside and the inside. That's why it's in the middle. The vessels are in between. Okay, and this vascular humor, or the vascular layer, is also called the uvea, and it contains different parts. It consists of different parts that I will talk about. But basically, you also need to know that it is dark. It's dark. Why is it dark? Because I want the light rays only go through the cornea. I don't want it to go from anywhere else, just like the camera. Okay, in the camera, you want the light rays to go through the front part, the lens only, right? You don't want it to go from anywhere else. And that's why I'm talking about the old-fashioned camera, not the phone, right? The phone, the camera, all of this, it has to be dark. It's black, right? All of this. Just in the front is the one that's open. So the vascular layer is also dark. So look at this picture right here. I talked about the fibrous, which is sclera, plus the cornea. Together, this is called the outer or the fibrous tunic. Look at the next one. You see three layers here? 1, 2, 3. Can we see the three? Okay. So the outer one, it looks bluish, but it is white sclera. Here is the sclera, and this is the cornea. Next, here it is. So this middle layer here is three parts. What's the other name for the middle or vascular layer? Uvea. Yes. So the middle or the vascular or the uvea contain choroid, the main part, all of this. Choroid until this. Here you have the ciliary body and then the iris. All of these together, this is the middle one. Okay. So what's the middle layer of the uvea? Choroid, plus ciliary body plus iris, the colored part. So the iris is the colored part of your eye. Okay, it's this circular colored structure in the front. Is it completely blocked? No, there is an opening in the middle of it. What is this opening? Pupil. Pupil. It's exactly in the middle of the iris. The iris, do we know that this pupil, the opening, it can dilate and constrict? Did you go to the doctor before and you see how they dilated and constricted? It dilate and constrict because we have two types of muscles. This is the circle like this. There are muscles that goes like this. And you see this going out like this. When they contract, they will pull and they will make this opening bigger. And this is we call dilation of the pupil, when the diameter increase. There are also circular ones surrounding the center part. When they contract, they constrict. Why do I need to dilate and constrict? We dilate if it is dark. We constrict if it is bright, right? Because if you go like in the sun, you have to constrict it. Otherwise, the extra bright light can actually harm your eyes. So you have to make it smaller. If you go like in a little bit of a darker area, you have to dilate to see as much as possible, right? But what are the muscles? Radial muscles, we can pull out for dilation, and circular muscles for constriction, okay? When you go to the doctor, they give you the dilator one, so they can see as much as possible. So look at this. This is a pupil. You can dilate, you can constrict. To dilate, you just pull out. Look at these radial muscles, the dilators, they pull out. And these circular ones, when they contract, they constrict. Ciliary body is in between the choroid and the iris. What's the most important thing to know about this ciliary body? It contains muscles and processes. This is important to know. Muscles and processes. The muscles is the ones that can contract and dilate to change the diameter of the lens. And the ciliary processes, they will contain suspensory ligament to suspend the lens. I will show you the picture, but I want you to understand this. Let's look at the picture here, so just to make more sense. Look at this. Can you see this part here? Ciliary body. This is the ciliary body. It contains muscles. And then suspensory ligaments to suspend this lens. Ciliary body containing muscles, suspensory ligaments, and the lens. So why the ciliary muscles are important? Because when these muscles contract, you change the curvature of the lens. Like my glasses or your glasses or any glasses, it's a lens, right? You can change the convexity. It can be like this, can be like this, right? It's different. So how can you do this? How can you do this or that? When you contract, you can do this. Relax, it goes like this. Do we need to change the curvature and convexity of the lens? Of course, yes. Because look at this visual axis. When you see something, the light rays come in, pass through the cornea because it is transparent, and then it passed through aqueous humor. It's watery, transparent. That's okay. Lens, transparent. Go through it. Even the vitreous humor in the back, transparent until you go to this point. What if I'm looking at something that's closer? Is it going to go to this point, which is the purpose or the target? No. What if it is too far? No. So depending on if you're looking at something that's close or something that's far, you need to change the convexity or the curvature of the lens by these muscles. So this is for the ciliary body. The choroid is that part that contains the blood vessels. I talked about it. It supply the outside and the inside with blood. Why do you need the blood? Oxygen and nutrients. I think everybody know that. The inner one is called retina. The middle one is called uvea. The inner one is called retina. And this is two layers itself. Pigmented one, this is the one that absorb the light. Neural one. What's neural means? Neurons, right? Nerves, neurons, yes. So this is the one that contain the actual nerves, the neurons. So it contains photoreceptors, photoreceptors, the visual receptors. And these receptors are two types, rods and cones, and or cones. This is an important information here that you have to know, that rods looks like a rod, something like this, looks like a rod shaped. And this is for vision in the night, dark. Very important. Rods, you use it at nighttime. At nighttime, when you look, you can barely see, right? If it is dark, you can barely see. You don't see colors, but you see colors in the day or if there's light, right? Right now, if I look at any one of us, I can see the colors, right? If you turn off the light, I won't see it. So the rods are used at nighttime and it's black and white. The cones, on the other hand, you can see colors, so it is obviously during the day. Is that clear? And if you forgot it, cones start with C. This is for the color. C, C. Cones, color. So it's day. Rods, black and white, night. You have to know this. So the retina, the inner one, the neural part of it, does it all contain receptors? Is it all the same? Not exactly. Not exactly the same. Okay? The central part contain more cones and the outer part contain more rods. Okay? There is a specific point that contain huge amount of cones. And this is called fovea centralis. Fovea centralis, very important. Fovea centralis. What's the fovea centralis? Part of the retina, a point in the retina that's heavily clustered, contain huge amount of cones specifically. Okay, and this is your focal point. So when you see something, I told you that you change the curvature of the lens, right? Why? So that it go to that point. What is that point? Fovea centralis. Why did you call it fovea centralis? It's at the center of something called uh, macula. Okay, in the center of macula. I will show you the picture, but look at this. If this is the neural inside, okay, we have a lot of receptors all over. Same, no, rods more outside, cones are more inside, okay, toward the center. In the part that contain the cones, there is a specific spot, macula, at the center of which we have fovea centralis. What's the most important here? Fovea centralis. Okay, you have to remember it. It's heavily clustered with cones, and the visual axis should go to that point, and that's why we call it the focal point. Okay, so when the light comes in, you need it to fall specifically in that point. What if the light goes into another point? You will not see clear. You will see just uh, hazy. The picture will be hazy. So you wanted the light to go exactly into that point. Fovea centralis contain a lot, and you can easily see. Now look at these. This is how the cones look like a cone, and these looks like rods. From there, both of them, when the light fall on those, you make an action potential just like anything else. From there, it will go to bipolar cells, to the ganglion cells, and these ganglion cells, its fibers will make the nerve, optic nerve, the nerve of the vision. Look at this picture right here. Do you see this circle? Look, like rods and cones all over the place. Outside here, what do I have more? Rods. What do I have more here? Cones. Clear? What is this circle? Macula. What's in the center of the macula? Fovea centralis. Cones. A lot of cones. Yes. Oh, okay. I'm sorry. Okay. So we'll stop here at the fovea centralis and we'll do the rest next time.